Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing Odisha CPET Microbiology question paper for conducted in the year 2020. Name the scientist who proposed the domain concept in biology. So it is Carl Woese. Answer is option C. One of the following genus belongs to Archaebacteria. So out of this it is Halobacterium that belongs to Archaebacteria. During gram staining of bacteria, iodine is used as. So the mordant in gram staining is grams iodine, and this binds to the crystal violet, making a large complex that adheres to the cell membrane. So iodine is used as a mordant in gram staining of bacteria. Answer is option B. The next one. In a microfungal cell, the ribosomes are, so they are 80S type. Microfungal is a eukaryote. So answer is option A, that is 80S. Then germ theory of disease was proposed by. So Louis Pasteur published germ theory of bacteria and proved that bacteria caused diseases. And this idea was taken by Robert Koch in Germany, who began to isolate specific bacteria that caused particular diseases. So out of these options, Robert Koch is more correct. Then next one. How many endospores are produced per single bacterial cell during endosporulation? So endospore, it is a dormant and non-reproductive structure formed under stress condition in bacteria. So under favorable conditions, it will germinate and reproduce. And the endospore, it contains DNA, ribosomes and dipicolinic acid. So how many endospores are produced per single bacterial cell? That is only one is produced. So answer is option C. Then next one. A bacterium having a tuft of flagella at one end of the cell exhibit. It exhibits lophotrichous flagellation. Answer is option A. The next one. Antibiotics synthesized by microbial cells are example of. So antibiotics are secondary metabolites. So answer is option C. The next one. When a plasmid in a bacterial cell gets itself integrated into bacterial chromosome, it is called as an episome. When plasmid get integrated into bacterial chromosome, it is called as an episome. Answer is option B. Then next one. The device that is used for continuous culture of microorganism is called. So in a chemostat, the bacterial population can be maintained at a constant density. So it is a type of continuous culture of microorganism. So chemostat is the device. So answer is option D. Then in a solid agar microbial culture medium, agar act as. So agar is a solidifying agent for microbial media. So answer is option D. The next one. The end product of biological nitrification. So nitrification is oxidation of ammonium to nitrite and then to nitrate due to the action of soil bacteria, archaea or even fungi. So answer is option C, that is nitrate. Then chlorobium species is. So chlorobium is a symbiotic green sulfur bacteria that perform oxygenic photosynthesis, and it function as an obligate photoautotroph. So the answer here is it is green sulfur bacteria. Answer is option C. Then one of the following is not a nitrogen fixing microorganism. So it is Rhizopus. It is fungus and it is commonly called bread mold. So answer is option B. Then the bacteria found in Saltern is. So in Saltern is an area of installation for making salt. So the bacteria that will be found will naturally be halophiles, halophytic bacteria. Answer is option D. Then next one. One of the following is the characteristic feature of photosynthetic green and purple bacteria. So the characteristic feature is that for absence of photosystem 2. Answer is option A. But there is presence of photosystem 1 in green and purple bacteria. Only photosystem 2 is absent. Coming to the next question. A bacterium having a generation time of 5 minutes is introduced into an appropriate culture medium to enter into log phase. What will be its number after 30 minutes? So we have the formula 2 raised to number of generations into initial number of bacteria is equal to final number of bacteria present after n generations. So the final time is 30 minutes. So in 30 minutes the bacterium will divide 6 times. So number of generations will be 6. So 2 raised to n is equal to 2 raised to 6 and that is equal to 64. So 64 into 1. Initial number of bacteria is 1 because they have given a bacterium having. And the final number of bacteria present after n generations will be 64. So answer C is equal to answer C is the correct option. So it is 64. Then next one. Which of the following steps during electron transfer in anoxygenic photosynthesis is the ATP production step? So it is anoxygenic photosynthesis. So in that it is from cytochrome B to cytochrome F. The energy released in electron transfer between cytochrome B and F is used for photophosphorylation, that is generation of ATP from ADP and inorganic phosphate. So cytochrome B to cytochrome F. Then which of the following structure is found in bacterial cell? So it is mesosomes. Mesosomes are infolding of bacterial cell membrane and they are analogous to eukaryotic mitochondria and they have enzymes associated with respiration. Then next one. Microbes which uses inorganic elements are reductant they are called as lithotrophs. So lithotrophs are organisms that use inorganic substrate to obtain reducing equivalents for use in biosynthesis or energy conservation via aerobic or anaerobic respiration. So it is lithotrophs. Then next one. Which pathway of glucose dissimilation do yeast carry out during alcohol fermentation? So it is EMP pathway. Answer is option B. Then next one. Name the electron acceptor during nitrate respiration. Nitrate respiration. It is nitrate. Answer is option C. Then which of the following disease is caused by a virus? So it is measles. Measles it is caused by a virus of the paramyxo family. Then tetanus is caused by bacterium Clostridium tetani. Meningitis is caused by viral or bacterial infection. And dermatitis because of contact with something that is will irritate the skin or triggers an allergic reaction. Then next one. One of the following is usually the skin inhabitant. So it is Staphylococcus aureus. Answer is option D. Then the bacterial species involved in yogurt preparation. So yogurt is a fermented dairy product produced by lactic acid bacteria. So the answer here is mixed culture of Lactobacillus bulgaris and Streptococcus thermophilus. Answer is option D. Then which of the following can be used as biofertilizer? So nostoc and anabena. So answer is option C. Then tyndallization refers to. So tyndallization or fractional sterilization is a physical sterilization method and it involves heating the material at 100 degrees Celsius for 20 minutes or on 3 consecutive days, intermittent with incubation at 37 degrees Celsius. So as per the options, option A is correct, that is heating with incubation. Then next one, the process by which all microbes are either destroyed or removed from an object is called. So what is sanitization? Sanitization is the process in which pathogenic microorganisms are reduced in number so that they are no longer harmful. And disinfection eliminates or removes many or all pathogenic microorganisms except bacterial spores from inanimate objects. And what is antisepsis? Antisepsis is the destruction or inhibition of microorganisms on living tissues, thereby limiting or preventing the harmful results of infection. What is sterilization? It refers to the process in which all living cells, spores, viruses are completely destroyed or removed from an object. So as per the question, that is the process by which all microbes are either destroyed or removed from an object, it is called as sterilization. Answer is option C. Then next one. During lactic acid fermentation by lactic acid bacteria, one of the following act as oxidant. So lactic acid fermentation by lactic acid bacteria. Which one act as oxidant? So pyruvate act as oxidizing agent in the production of which results in the production of lactate. So answer here is option C, that is pyruvic acid. Then carbon, nitrogen, phosphorus, sulfur, potassium, magnesium, calcium and sodium. Which of these is a micronutrient or for microorganisms? So the answer here is option A, that is sodium. Then next one. In a microbial culture, when all the growing cells are in the same stage of division, the culture is called. So it is called as synchronous culture. Answer is option B. Then phenol, dettol and chloramines dash which one is the disinfectant? So phenol is the disinfectant. Answer is option A. Then one of the following is not an antibiotic. So tetracycline, amoxicillin, bacitracin, sulfadiazine. So as per me, all are antibiotic. If there is any doubts with this particular question, or you have got a different answer for this particular question, then you can give it in the comment section. Then pasteurized milk samples are. So pasteurized milk samples, they are pathogen free. They are not sterile. They are pathogen free. Answer is option B. Then aflatoxins are, they are produced by certain fungi that are found on agricultural crops such as maize, peanuts, cottonseed and tree nuts. So they are mycotoxins. Then next one. Sugar constituents of bacterial wall peptidoglycan are. So the sugar component consists of N-acetyl-glucosamine and N-acetylmuramic acid. So it is amino-acetylated glucose. Answer is option A. The next one. Virus particle present outside the host cell is called. It is called as virion. Answer is option B. Prophage, the viral DNA get integrated into the bacterial chromosome and it replicates along with the bacterial genome. This integrated viral genome is called as prophage and it is relatively harmless and continues to remain in that position until unless lysis occurs. Then viroid are the smallest infectious pathogens comprised solely of a short circular RNA without protein coat. Then virusoids are single-stranded RNA that require other helper virus to establish an infection. The next one, the viral genome when remain integrated with the host bacterial genome is called. So it is called as a prophage. Answer is option A. So lytic phages, they take over the machinery of the cell to make phage components, and then they destroy or lyse the cell, releasing new phage particles. Then what is progenote? It is a hypothetical stage in evolution, any primordial organism in which relationship between genotype and phenotype emerged. That is called as the progenote. Then next one. One of the following genetic exchange in bacteria involves physical contact between bacterial pair. So that is none other than conjugation. Answer is option D. As per Burgess Manual of Systemic Bacteriology, the phylum Proteobacteria comprises of. So the phylum Proteobacteria, it consists of 5 classes: alpha proteobacteria, beta proteobacteria, delta proteobacteria, gamma proteobacteria, epsilon protobacteria. So, totally 5 classes. Then name the domain to which cyanobacteria belongs. So it belongs to eubacteria. Answer is option C. What is the function of an impeller in an industrial fermenter? It is for mixing the content. Answer is B. Then, for the industrial fermentation of glutamic acid, which bacterial genus is employed? It is Corynebacterium glutamicum. Answer is option A. What is the photosynthetic product of blue-green algae? It is cyanophycean starch resembling glycogen. Then, which is not true for the heterocyst of nostoc? Intercalary position, yes. Site of nitrogen fixation, yes. Presence of polar nodule, yes. Carbon dioxide fixing ability. So carbon dioxide fixing ability is not true, is not correct with regards to heterocyst of nostoc. The next one. One of the following can be used for sterilizing microbial growth media. So it is microbial growth media, so you can use an autoclave. The process of making beer is called brewing. Answer is option B. One of the following is an acidophile. It is Thiobacillus ferroxidans. Answer is option A. Then nitrate respiring bacteria they are anaerobes. Answer is option B. One of the following is not a prokaryote. Answer is Candida. Candida is a fungus. It is a eukaryote. Resolving power of microscope is a function of the wavelength of the light, refractive index, and numerical and angular aperture. So as per me, A and B are correct, but not numerical aperture. A and C are correct. As per me, A and C are correct, not A and B. It is not numerical aperture, it is angular aperture. Then, in which specific growth phase the recipient bacterial cell develops competency to take up donor DNA during transformation? So it is the late exponential phase. Answer is option C. Name the enzyme that act on the DNA after its entry into the recipient cell during bacterial transformation. So after DNA entry into the cell, one strand is immediately degraded by deoxyribonucleases, while the other strand it undergoes base pairing with the homologous portion of the recipient cell chromosome. So they have asked name the enzyme that act on the DNA after its entry into recipient cell during bacterial transformation. It is deoxyribonuclease. Then next one. HFR bacterium conjugates with the F plasmid containing bacterium. What will be the type of bacterium after F minus it is? Okay, F minus plasmid containing bacterium. What will be the type of bacterium after the conjugation is complete? So it will be F minus itself. Why? Because HFR is F factor integrated into bacterial chromosome is called as HFR. So in mating between HFR and F minus cell, the F minus cell never becomes an F plus cell, because during the the F factor in HFR is nicked in the middle during the initiation of strand transfer. placing part of the F plasmid at the beginning and another part at the end of the strand to be transferred. So to become F plus, the HF or HFR, the recipient cell must receive the entire F factor. But this does not happen, and before that the conjugating cells, they will break apart. So the F factor is not transferred completely. So the cell remains F minus itself. So answer is option B. Then next one. Yeast extract usually employed in bacteriological culture media because it acts as the source of. So it acts as a source of vitamin and micronutrient. Answer is option A. Then which of the following microbial product is used as bioplastic? It is polyhydroxybutyrate. Answer is option C. Then Bacillus thuringiensis is used as, so it is used as an insecticide. Answer is option A. Which of the following is a rich source of protein? Spirulina, chlorella, semidesmus. All of the above. All are rich sources of protein. Then retroviruses like HIV which causes AIDS are RNA viruses. Then which of the following is a single-stranded RNA virus? So SARS coronavirus, it is single-stranded RNA with positive sense. Rotavirus is double-stranded RNA, and hepatitis B virus, it is circular double-stranded DNA. So the answer here is option A. Then capsid of a virus constitute. So it is made of capsomeres. Answer is option C. Which clinical sample is used to diagnose hepatitis B? So a laboratory diagnosis of hepatitis B infection is dependent upon the detection of hepatitis B surface antigen in the serum. So the answer here is option B, that is blood. Enveloped viruses matured and released by. So they are enveloped viruses. Okay, so it is budding from cell membrane. Answer is option C. Who is considered father of virology? Martinus Beijerinck. Answer is option C. Pox viruses are. So they are enveloped DNA viruses. Answer is option A. Icosahedral structure of virus has. It has 20 phases. Answer is B. Then the virus of family Baculoviridae infect insects. Option D. The structure of tobacco mosaic virus is. It is helical. Answer is option A. Then industrial fermentation of vinegar involves. So vinegar is the product of two-stage fermentation. In the first stage, yeast converts sugar into ethanol anaerobically, while in the second stage, ethanol is oxidized to acetic acid aerobically by bacteria, Acetobacter and Gluconobacter. So the answer here is yeast and acetic acid bacteria. Option C. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.